Okay, hey everyone, JP here. This is the last actual section for proteins and peptides. So this is part 3E. We're going to talk about unnatural amino acid incorporation. And specifically what this is going to talk about is how we can incorporate unnatural amino acids into proteins using the biological machinery. All right, so this will be, as I mentioned, the last unit of this section. And before we talk about unnatural amino acids into proteins using the biological machinery, let's first talk about, why do we have only 20 amino acids? And let's look at the genetic code a little bit to see, if we can take advantage of some of the redundancy. So if we look at this picture from molecular biology, it shows that there are 20 amino acids. We have 20 amino acids, but we actually have 64 different potential codons. So, so you've learned, you can read this like UUU corresponds to phenylalanine, but UUC also corresponds to phenylalanine. So why do we have this redundancy? Well, there's some postulated reasons. One is, well, if there's a mutation, then this acts as a safe check so that you won't get a change in amino acid necessarily. And you can see that different amino acids have different degrees of redundancy. So phenylalanine, there are only 2 sort of codes for that, whereas leucine actually has 6 different ways to code for that amino acids. Methionine, on the other hand, only has 1, right? So there's different degrees of redundancy. They're not created equally. So maybe that's one reason. We have 20 natural amino acids. We also have 3 different stop codons, all right? So whenever the message is supposed to stop and the peptide is to be released through, different elongation factors from the ribosome, a stop codon is reached, okay? And we have 3 different stop codons. We have UAA, we have UAG, and we have UGA. Okay, now it turns out that the one that's used almost exclusively 95% of the time is UAA. These other 2 stop codons, they're valid, but they're actually used rarely. And, we could think of why that might be the case. It could be sort of a safe check in place. It's always good to have, if you're driving your car, for example, you have your primary brakes, right? But rarely do you actually stop the vehicle with your emergency or parking brake, right? But it's good to have just in case. So biology kind of employs that same type of strategy. So, okay, so this is interesting. We have actually 64 different potential codons, so we could potentially have 63 different amino acids in one stop codon, right? But nature's said, no, let's keep it simpler and let's put some checkpoints in place. Fair enough. However, if we are trying to expand the genetic code or expand the type of amino acids available to a protein, then we need to look at this chart right here and say, well, where could we add a 21st amino acid? All right, so this UAA is used 95% of the time, but UAG is rarely used. This is actually called the amber stop codon. Okay, so maybe you can think of it kind of like an orange light or something. So since it's rarely used, could we evolve a unique codon tRNA pair and corresponding amino acyl tRNA synthase for this amber stop codon? Could we find a way to actually use it since it's just so infrequent? So this is exactly what some researchers at Scripps Research Institute did. So this was led by Peter Schultz. And basically what they did was they took a tRNA molecule, and this tRNA This molecule codes for the amber stop codon in the anticodon loop. And attached, or what is actually holding the tRNA to this protein is called a tRNA synthase. And so this loads up the tRNA with the amino acid, and we learned a lot of this during the biological synthesis of peptides. This is the activation site here. So if you're using a different amino acid R group that's unnatural, you could modify this pocket here to accommodate that larger amino acid. So this active site here can be modified to accommodate the new amino acid. And then the anticodon loop, this codes for UAG, the stop codon. Okay, so kind of looks, you know, somewhat simple. I mean, on paper, you know, it is simple. You can evolve a new tRNA, you know, synthase with an activation site that's altered to accommodate an unnatural amino acid. This is another representation of how this would actually look, unnatural amino acid incorporation. So under standard conditions, this would be your canonical amino acid with an endogenous tRNA. The endogenous synthase comes in, adds that amino acid to the tRNA, then you get your standard triplet codon base pairing with your standard amino acid. So in this case here, we're going to use an orthogonal synthetase, place in a natural amino acid, and the synthase will have to be modified, as I mentioned previously. An orthogonal tRNA that would have a unique codon pair, and in this case we're going to use that very rare codon called the amber stop codon. So what this means is that you can genetically engineer an mRNA sequence in where you want to put in a natural amino acid. You simply have to add the amber stop codon. So if you didn't do this unnatural amino acid bit here, then you would get premature stoppage or premature cleavage of the peptide. However, with this orthogonal synthetase and orthogonal tRNA natural amino acid, you're going to have a protein now that can be made that now has the unnatural amino acid. So how does this work, right? So I've kind of talked about, you know, what this would look like sort of at the end, but how would you actually do these experiments? All right, so this is all a lot of chemical biology, and this is going to be your cell. And so you're going to need to feed your cell, you know, we'll just call this the unnatural amino acid. So whatever the R group is, and really whatever that stereochemistry is, it could be pointed to you or away from you, really depends what your sort of desires are. So you add that to the cell. Now the cell needs to have two components to it. It needs to make that tRNA that has that amber stop codon. So it's a modified tRNA. So we're going to have a plasmid here. Call this plasmid 1, and it encodes for the modified tRNA, the amber stop codon, and mutant tRNA amino acyl synthase. Okay, so you got to have the synthase that's going to bind to that tRNA. Okay, and you know, I'm really simplifying this to find a mutant tRNA amino acid synthase that would accommodate the amino acid and bind to a modified tRNA, that takes multiple rounds of selection in order to get that specificity. Okay, so once you've optimized a plasmid, this process here to generate that plasmid could very well take years, right? So let's assume that we've already done that work, or we have that already to our disposal. Number two will be plasmid number two, and this is going to contain your protein of interest that you are interested in. So this will encode the protein and the mRNA of interest with the amber stop codon. Okay, so encodes, well, it's a DNA plasmid, okay, but encodes the sequence for the protein of interest, and obviously the mRNA must have with the amber stop codon. Okay, the amber stop codon is placed at the site of interest for the unnatural amino acid incorporation. Okay, and then, so you take those three components and you let your cells grow, right? And then you're going to have now multiple copies of your cell, and what's going to happen is you're going to generate your protein, and your protein is going to now have that amino acid attached to it as a star. And then this protein can then be lysed from the cell, purified, and you can do whatever experiments you tend to do with that. And during this process, I want to say what kind of different amino acids can we add, right? So we have a variety of different amino acids available. We have 20, but I just want to highlight a table of unnatural amino acids that have been biologically incorporated into proteins. So you can see here that we have a wide variety of different structures. Okay, it might be a little tricky to see. I'll see if I can zoom in maybe a little bit. So if you look closely, like you can see that we have very different groups here. We have an allyl group here. Here we have some nitro groups. These are unnatural groups. Here we have a nitro like phenyl group here, which is often used as a light labile protecting group. So you could, incorporate this and then this would get removed. Down here you can see you have like a iron metallo type of amino acid. So you could add, for example, iron to a protein. This here is a molecule called azobenzene. So you have two nitrogens here held together by double bond. And this is an interesting modification because in the presence of UV light, this will actually flip to cis. And what it does, it actually changes the structure of that particular molecule. So this will actually flip to cis from trans, and then visible light can revert it back. So you could actually have a molecule that adopts one conformation, like a protein, in the presence of UV light, and a different one in the presence of visible light. So you could potentially alter the structure and function of that protein and control it. So I'm not going to go through all these, but you can see that there's just an enormous wide array of different types of functional groups. This one here is called benzophenone. I'll just zoom out a little bit. Benzophenone is also quite interesting because in the presence of light, you actually generate this radical at the central carbonyl. And this radical actually will covalently bond to nearby proteins as a radical. So So this is actually really useful if you're trying to understand the mechanism of where maybe your protein is binding at a particular spot, because then you can map out using maybe mass spec or even through degradation and looking on gels where the protein's binding. Okay, so why bother with unnatural amino acids? So we've talked about the system, we've talked about how we can put one in, but I've already taught you that you can actually, just using solid phase chemistry, you can actually add in unnatural amino acids just using solid phase chemistry. In fact, you can make peptides, or you can make things that look even much different. So why would we want to bother with this approach? So of course, if this were a regular class, we would have some sort of discussion. However, one big advantage is that if you recall with solid phase protein or peptide synthesis, you were limited to the size of the peptide. And even though you could do perhaps native chemical ligations, maybe once, twice, there are a lot of proteins are much larger, or you need to look at the whole entity. So this advantage allows you to use the protein in its native form within the cell as well. Okay, so advantage. You can track the function of the protein. You can use these unnatural amino acids within the biological system of the full protein. Another thing too that sometimes happens when you're making peptides or proteins in solid phase synthesis is that you might be able to make it, but it might not fold correctly to its proper form. It needs to fold correctly, and this is assisted by some proteins called chaperones to help it fold correctly. And sometimes with those chaperones, it's not going to fold the right way, and then therefore you might have a protein that doesn't function properly. So the advantage is you can track function, but it allows the protein to exist in its native state. And it allows for long proteins to be made as well. You can also track the function within biology, right? So you could potentially put amino acids like, fluorescent amino acids, and you could actually perhaps ask questions like, where does it go in the cell? What is its mechanism of translocation? Does it build up somewhere? What is its half-life, right? So you could track the proteins in cell. It'd be very difficult to do that if you made the protein by itself and sort of try to introduce it artificially. Another interesting thing too is, and again, these are just in addition to some of the amino acids I showed on the previous page, you could use D-amino acids. And this is also very useful for examining, enzyme sites or the mechanisms of how particular enzymes work. So sometimes it could give you an idea of how stereospecific these reactions are that an enzyme works, or maybe it's not stereospecific. So it can give you really some insights in terms of enzymatic mechanisms. Okay, it is, it is actually, I've never done it myself, but I've read and I've heard that it is actually a cumbersome process, and it's not so easy. Okay, so generally speaking, when researchers need to add in a natural amino acid, there's two main ways, right? There's the amber stop codon method that we just learned now, and then there's a chemical synthesis method that we talked about a few weeks ago. Okay, so we're going to do a little comparison of the amber stop codon method compared to chemical synthesis method. All right, so advantage of amber stop codon method, the advantage is that you can have an unnatural amino acid can be inserted in vivo. And in theory, there really is no restriction on length. No restriction on length of that protein. And you can study the protein in its native biological environment. Okay. Some disadvantages. Okay. Because, we have 64 different codons, but we're only using the amber stop codon. This allows for only one type of a natural amino acid per protein, right? You can't start using some of the other codons, because then you're going to start modifying them at many different places. So only one type of a natural amino acid generally can be used. It is very cost and time consuming, and sometimes the yields are just maybe not so great. Like, like you're trying to put this on natural amino acid, maybe the selectivity specificity is not always there. You might get low yields, whereas with sulfate synthesis, you can really purify that protein and generate pretty good quantities of that. And sometimes the chemical modifications, maybe they could be toxic to the cell, right? If you're using maybe some very sensitive functional groups or metals or something, who knows, right? Okay, so now we're going to compare this to the chemical synthesis. So in general, there's no restriction on the number of unnatural amino acids. I mean, you could have a peptide protein that you make chemically that is made entirely of unnatural amino acids. As I just alluded to earlier, you can, like, large amounts of purified protein, of modified proteins can be generated. Whereas if you're dealing with a cellular system, the yields might be low, and it might be actually quite challenging to get just your isolated protein from the cell, if that's what you're trying to do. Okay. Disadvantages. Well, the biggest one is the restriction on length. Okay, so chemical synthesis of peptides is restricted depending on the length of your peptide. You can't make 1,000 mer protein using chemical synthesis. It's also difficult to it's difficult to monitor true in vivo effects due to permeability and proper folding. So, if you add unnatural groups to peptide proteins, it's hard to say whether, inactivity or activity is real, perhaps, because you're making assumptions that it can fold properly, that it can get inside the cell properly, etc. Okay, so these are the main advantages and disadvantages. Certainly there could be more, and we could think of having a discussion about that. But essentially this wraps up the whole protein section of this course. So I hope you find these videos, these modules, some of them are a little longer than others, some are shorter. Hope you find these useful, and you can definitely view them as many times as you want and need to. Okay, so that's it for now. See you next time.